Okay, so today I will be teaching you guys about the lac and trip operons. The lac operon is a cluster of genes that contain the DNA sequences to regulate the metabolism of lactose. The lac operon consists of a promoter, an operator, a terminator, and the genes lac-Z, lac-Y, and lac-A, and also has a cap site, which is a catabolite activator protein, here at the start. Essentially, it all depends on the environment and the conditions that the actual operon is in, which are listed right here. So in a low lactose environment, it's going to have no transcription. This is because it doesn't actually need the enzymes that break down lactose. In the low lactose environment, there is a repressor binding to the operator, preventing the RNA polymerase from moving down the DNA strand and making and coding for the enzymes. However, in a high lactose environment, the cell is going to want transcription. This is because there's allolactose, this little guy right here, in the environment lying around, which binds to the repressor, which moves the repressor molecule off of the operator. Therefore, the RNA polymerase is able to move along the DNA sequence and code for lac-Z, lac-Y, and lac-A. The gene product of lac-Z is beta-galactosidase, which cleaves lactose into glucose and galactose. Lac-Y encodes beta-galactoside permease, which is a membrane protein that becomes embedded in the cytoplasmic membrane to enable the cellular transport of lactose into the cells. And lac-A encodes galactoside acetyltransferase. When the RNA polymerase reaches the end of the operon, it approaches the terminator region, which is this region right here, which actually dismantles or breaks apart the RNA polymerase from the DNA strand, and the RNA polymerase will rebind back to the promoter to continue the cycle. The other part of the lac operon is the cap site, or the CAP site, which actually takes place in glucose environments. So in a high glucose environment, there's low CAMP, and in a low glucose environment, there's high CAMP. So if there's not a lot of glucose, this means that there's not a lot of energy the cell can use. Essentially, an environment with low glucose will have high CAMP, which means the CAMP, this guy right here, will bind to the CAP, just like that, which will then bind to the CAP site and increase transcription. Following that, this is an example of the trip operon. The trip operon is basically the operon that codes for enzymes that create tryptophan, which is an example of biosynthesis. So in a low tryptophan environment, the cell is going to want transcription because you're going to want to create tryptophan since there are low amounts within the environment. However, if there's a high amount of tryptophan, this one right here, there will be less or no transcription since you do not want to create more. Ultimately, in an environment where there is lots of tryptophan, the tryptophan acts as a co-repressor and binds to the repressor molecule, just like this, and the RNA polymerase goes on the promoter, which is so the co-repressor binds to the repressor, and the repressor is binded to the operator. Ultimately, this allows the RNA polymerase to move along the operon to code for the genes as the repressor moves off of the operator and moves all along the operon to code for the genes. However, in an environment with low tryptophan, there will not be a co-repressor. Be like that. Therefore, the repressor cannot bind to the operator, and the RNA polymerase can move freely throughout the operon again. And code the genes, creating the mRNA, and then again being translated into polypeptides, creating the enzymes that will create tryptophan. Thanks.